One of the things we've discovered, I know some of you have gotten emails from me saying that you hadn't watched a video yet, and you're like, yes, I did. I watched it 3 times, as a matter of fact. First of all, when I send those emails, it's like a mass email. So out of Google Classroom, I just hit this button that's anybody who hasn't turned it in. And then it sends an email to those people. So don't feel like I'm like personally picking on you. I just select it all, and it's more just to remind you guys, anybody who needs a reminder. So something I recognized today when I was writing up my agenda is that I need to move a little bit faster with you guys. Normally we spend just less than 2 weeks on this unit, and now we're, you know, finishing up the 3rd week and we're not done. And that's not because you guys aren't capable of moving faster. It's because I haven't caught on to how this pace is different. Like normally I would introduce carbohydrates on one day and then lipids the very next day. And I have to remember that these Mondays and Thursdays are actually two periods. So like on a Monday, if we were in class, we would do our carbohydrates, and then I'd have follow-up activity related to carbohydrates. And that's what I've been doing with you, but really I'm filling 2 day slots that way. So I realize now what I should have done was introduce the carbs in the day, and then you worked with lipids in the afternoon, or, you know, in your flex time. Or I did carbs and lipids, just shorter versions. I don't know. I'm thinking that maybe my lectures, I might have to turn, like you've watched a couple videos, you watched the proteins, and you're going to watch the nucleic acids next. And maybe I push my lectures out earlier, and then we can just work with the pieces that we struggle with in class, because you know, in a short Zoom, we don't have a lot of time for both presentation and problem solving. So I'm going to try to like alter my way, because see here, we're supposed to spend 5 to 7 class periods on this unit, and you got to count Mondays and Thursdays as 2 days. So anyways, if we add it up, it's a bit extra days. And if I keep going like that, like it's not a problem right now, but if I keep going like that, come May, we'll be definitely out of review time, and we might be short with our last unit. So today I wanted to talk about protein structure. So I just want to clarify the thing. So hopefully you watched the lecture, and I think protein folding is the most difficult part. So I'm going to talk about protein folding, and I meant to have some manipulatives. In the flex time, you're going to do a pattern matching. It's a PowerPoint, and you're going to manipulate. So you're going to move things to the right categories, and that's going to be all 4 macromolecules, not just proteins. And then I'm going to have you do the nucleic acids video on your own, you know, sometime between now and Monday. And then I would like to do a lab on Monday, and we'll test on Wednesday. So I haven't decided yet if I'm going to like go in person and do the lab in the lab, and then video pieces. I did find like a simulated lab online. So I might video what it really would look like, but push out the simulation for you guys to do. So put on your calendar, next Wednesday, we'll do the test over Unit 1, macromolecules. I will post, I'll push out through the AP Classroom, the Unit 1 progress test. So right now you guys have watched all these videos, and what we've determined is that if you don't watch the video all the way to the end, it doesn't mark it as done. So that's why maybe if you've watched it and I'm saying AP says you haven't watched it, maybe you're like clicking it off. So just let it play out to the very end, and then it should show up as being done. So when you go to your AP Classroom, this is the home page when you click on the home tab, and there's all the videos we're supposed to watch. You can find them under the units tab. So like it comes up to whatever this tab is, and that's just an overview and it doesn't say anything. So if you click on the unit, if you're struggling for the videos, to find on your own rather than using my links, that's where they would be. Under your assessments tab, I see that work 3 topic questions part 1. So we needed to have the lockdown browser, and in the Google Classroom with Monday, because that's the day we did this, I pushed out directions for putting the lockdown browser on your computer. And you can put it on your laptop if you want, like there's different directions, or the first page tells you how to put it on the school issued Chromebook. So if you want to put it on your Chromebook because you don't want to put something foreign on your own personal computer, then do that. And then when we do those practice questions or assessments, we can go onto the Chromebook for that. So you'll find Monday was the day that we struggled with that. So I put the directions on Monday. They're like attachments for you to open. So this is, when you look at Unit 1, these are all the things we should have been studying or read about. And we don't have time to cover everything. Like I didn't cover endergonic and exergonic reactions, kind of hoping you read about that, and it's kind of easy piece. So hopefully you understood it. If you didn't understand it, any of those things you need to go out and find resources for. It's in your textbook, so if we're reading our textbook, we're gonna come across it anyways. We are gonna hit enzymes by themselves next, so we're gonna go further into enzymes. But you'll also find it in the next couple of things I'm gonna show you. Okay, so we did water, and we talked about, we definitely wanna know the properties of water and how those properties allow water to do what it does. So we want to talk about makeup and properties of macromolecules. That's what we've been talking about, their functionality and how they work, how they're put together. We've covered the dehydration and the hydrolysis reactions. So enzymes, that'll come next. Anabolic building, catabolic breaking down. Endergonic is taking in energy, and exergonic is putting out energy. But like I said, I'm going to hit that after we do the next macromolecule, after we do our macromolecules test. And then we got to talk about structures of DNA and RNA, which you're pretty familiar with. So if you want to know like exactly what the College Board is looking for, if you click here, this isn't the big CER that you guys did the very first day of school. This is just related to Unit 1. And I also, like any of these links are also attached to today in the Google Classroom. So if you open that up, it'll go through and tell you specifically what you need to know about adhesion, specifically what you need to know about protein structure, that sort of thing. This is a review PowerPoint for Unit 1. So again, this is linked in Google Classroom. I'd open it, but I feel like it's gonna, take more time, and you guys can go through that on your own. But it takes each one of the essential knowledge pieces from the CER, and it'll point that out. It'll give you videos for review. So if there's something you weren't understanding, it'll point you in the right direction. It'll give you a little bit of tips and tricks, basically. So under the Google Classroom, all those things are linked. And this is where I said if I do this, it'll probably take time, but I feel like this is today, Zoom time, so much to do. So all these things that I'm pointing out, they're right here. So you can just open up the Zoom day and just click, 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 click if you want. Okay. So today, talking about proteins, looking at test questions, it's really important that you're able to give examples, like in an FRQ style, examples of different types of proteins. And, originally they thought, like, is it DNA or proteins that contain our genetic material? And the reason why protein was even a factor was because of how many different jobs the proteins serve in our bodies. They do so much. So it was fair to think they could possibly be our genetic code. So we know they're related to metabolism. Enzymes, most enzymes end in ASE, so we'll recognize that. Pepsin is like one of your digestive enzymes, so it's related to metabolism. which is both the buildup and the breakdown of, like it's the sum total of all chemical reactions going on in your body. Okay, so our next piece, after we take our Wednesday test, we're going to go into more details with enzymes. There is a like an AP College Board lab that we need to do that like all kids across the country are doing. So we need to be really familiar with that. As we go through the year, we'll look at how proteins serve as structure. So like for us, we might be familiar with keratin and collagen in our fingernails and in our skin. Collagen is in your ligaments and tendons. Transport, so in biology, we did talk about active transport and facilitated transport, where we used proteins that were embedded in the membrane to move molecules. And we'll talk more about those. This year, we're going to touch on the aquaporin, which is in your kidneys, but also like plants use it. It's when you need to move mass amounts of water. Antibodies with this characteristic Y structure. So they serve in defense, right, against foreign substances in our body. Proteins play a big role in regulation, and the AP loves things related to regulation and cell signaling. So when we get into that, we're going to be talking about the receptors and the signals. So like hormones, like insulin, receptors like acetylcholine is on your muscle cells. And when it, well, acetylcholine is a neurotransmitter that will bind with your muscle cells. And when it does, it causes these proteins, actin and myosin, to slide past one another. So that's actually what causes it to contract. So those of you who have had anatomy in the past or who are taking it now, or if you're going to take it in the future, you'll be familiar with actin and myosin in the next chapter for us. And then proteins also serve as storage. So I think our best example is beans. Like the seed itself contains protein that is stored energy, right? So we've already talked about how carbohydrates and lipids give us energy. Our last source of energy is our proteins. Like from our bodies, we're going to use up our carbohydrates first. And when we run out of that, we're going to use our lipids. And when we run out of that, we're going to break down proteins. So for us to use proteins, we're really in a state of like malnourishment. Okay, so this is the PowerPoint you guys did on your own. I just grabbed certain slides that I thought needed to be emphasized. Okay, so it might feel like that this is a quick transition and not a big flow, but you need to be familiar with the functions and give examples of. You also need to be very familiar with the structure, and this is where we start talking about the directionality of amino acids. So we've talked about our amino group. You hear that, Sophie? I said amino. Aren't you proud of me instead of amine? Amino group and our carboxyl group. So we talk about the N terminus and the C terminus end. So terminus is referring to the end. So it's almost repetitive in nature. So amino acids, because it has an amine group, and the carboxyl gives it an acidic property. So it's amino acids. Okay, the R group, remember that's some side chain, carbon, hydrogen, and it would have other functional groups attached to that hydrocarbon chain. So that's variable. Each amino acid, this is what's different. So we got 20 different amino acids. The blue, black, and green, all the same. What makes each one different is what we put in the R group. And then we're going to talk about how different R groups give that protein or that amino acid different properties. Okay, so this next one. So we can see the orientation here on the N terminus and the C terminus. This is where the bond will happen for the next molecule. Okay, so we'll see that on the next slide. Nope, in a couple slides, I lied. So this is talking about the R groups and how they play a role in the functionality. So remember, if something is nonpolar, it is repelled by water. And remember, our methyl group is nonpolar. So any of these amino acids that end with methyls are going to be nonpolar, and therefore they're going to be hydrophobic. They're going to fold away from the water. So proteins in a cell, your cytoplasm is all water, right? So they have to, so basically that's saying they're folding to the inside of the protein. And that's going to be really important as we go through protein structure, genetics, the denaturing of enzymes. So that plays a role many times as we continue on. The sulfhydryl does not have a polar property. So all of these are going to be nonpolar. And you don't have to memorize that. If you recognize the functionality of the functional group, then you can look at these structures and you can identify its property. So you'll see questions where it'll give you the structure. It's not necessarily going to give you your name. It may give you the name, but the name is irrelevant. It'll give you the structure, and it'll wonder where in the protein you'll find that molecule. Or if I swapped one of these out for a polar molecule, how would that change its property? So this is just showing you how they're folded inward. So the polar ones, remember hydroxyl is very polar. Amines are polar. So if we see any of those at the end in the R group, that's going to give it a polar property, and therefore it's hydrophilic, which means it likes the water. So it's going to fold outward to the outside of the cell. Or if it's in a membrane, it'll be at the outside of the cell. The protein will be to the outside of the protein, okay? So it's gonna be a surface. So here, like here's a protein that's within this membrane. You would expect the amino acids, because remember proteins are long chains of amino acids. You would expect the amino acids that are hydrophobic to be inward, the last slide. The hydrophilic are on the outside. And that's this slide, okay? Another thing that the functional groups can do is give it ionizing properties. Remember, ionic bonds are kind of like magnets, right? Positive and negative go together. So this is going to play a role in the folding of the proteins, where a negative R group will be attracted to a positive R group. So it's going to cause those to fold towards each other. So this is going to play a role in our tertiary structure when we get to that. And so if you look at a molecule, again, we don't necessarily have to memorize it, but these carboxyl groups, they can lose hydrogens, or they can tie up hydrogens. So if they're losing hydrogens, they play an acidic role. And if, I'm going to go back, sorry. And if they tie up hydrogens, they'll play a basic role. So if you see like a negative or a positive on a structure, you know that it's ionic. Okay, and it's going to need to attract other ionic molecules. So like when we talk about, a good example is substrates and enzymes at the active site. So remember, there's not going to be a covalent bond that holds a substrate to the enzyme, because that's a really strong bond. And you want to be able to release the substrate from the enzyme when it's done doing its work, right? So it's going to have temporary, like hydrogen bonds are temporary, ionic bonds are temporary. So if this is negatively charged and it's in a protein, I would expect, say this is my substrate, on the enzyme, it's going to be attracted to a positive amino acid in that enzyme. So I've seen questions related to that type of a relationship. So this is the one where if they're then accepting hydrogens, if they're tying up hydrogens, they're going to have a positive charge. So amine, remember, they can tie up hydrogens. That would be a good example. So here I see the positive on these amino groups. So that makes them ionic. It's going to play a role in tertiary bonds. It's going to play a role in temporary bonding to other molecules. Okay, so here I can see this amino group is going to be attracted to this carboxyl group, and that's ionic attraction. It also means, we'll get into that later. With denaturation, you can break those ion bonds. So covalent bonds are going to be between the amino groups, and take that away. They're between the amino acids, and that's the directionality. So here we see the amine O group is at the beginning, the carboxyl is at the end. every single one will be oriented the same way. So that gives the building of the protein a direction. Nucleic acids have the same concept where you can only build in one direction. So we would expect to see a bond between the amino group and the carboxyl group, okay? So this is showing you, is this going to be dehydration synthesis or is this hydrolysis? You guys want to throw that in the chat real quick? The fact that it's building should be a real quick, you know that that's a synthesis, and I see water is coming out of it, so that's dehydration. So lots of questions I've seen related to that. So I've also seen questions where they have these two amino acids in several different orientations and it says which one is correct. So that's where you're going to want to remember the carboxyl is at the end and it'll bond to the beginning, which is the amino. Any questions so far? Okay. So hopefully that all clicked with you when you went through it on your own. So this is the part that gets into the structure. And primary, secondary, tertiary could be confusing to us. So primary structure is just the sequence of the amino acids, right? So 20 different amino acids, depending on the order they're put together, they make the primary structure. So some people are not familiar with the little degree sign here. It's just like a short way of making the one into primary. So 2 with a degree is secondary, 3 with a degree is tertiary. When you get to college, your profs go way too fast for you to write everything down, so you come up with your shorthands, and anytime you have symbols, you'll end up using them. Like I never write reaction anymore, I just write RXN. But that's what the degree is. It's turning the one into primary. Okay, so primary is just your amino acid chain. You can kind of think of it, I mean, it can have any shape, but it's really just the linear shape or the linear sequence. And that's determined, you know, by the DNA. It's determined by the genes in the DNA. All right, so if we make any changes to this amino acid structure, we're gonna change the levels that follow, right? Because this is where I get, well, this level probably doesn't change. These two will change because it has to do with sulfhydryl bonds and ionic bonds and hydrophobic bonds. So if I change like one that was ionic, like if I change a polar for a nonpolar, or if I changed an acidic for a basic, it's going to have different reactions when I get to the tertiary level. So they'll fold differently. So if I change the primary, I affect the tertiary, which also affects the quaternary. So each protein has a unique structure. We look at this amino acid sequence, and we can look at relationship between organisms for evolutionary relationships. So the closer two organisms are, the less different their amino acids would be. So like the donkey and the horse, they have just one difference, but the horse and the chicken have 11 differences. And we know they're very different, right? So that's one of the things that they use when we get into DNA technology and evolution. That's one of the things they'll use to identify differences. Secondary structure, this is reactions between that hydrogen portion of the amino acids, okay? So you call this local folding, so highlight that. They either form beta pleats, where hydrogen bonds are folding together like this. So the hydrogen is attracted to the carboxyls, and so they form hydrogen bonds that way. Or they form helixes, and there's alpha helix and beta helix. It doesn't matter, but a helix is going to be a spiral. So this is interaction between nearby. So these are relatively close, right? And I'm going to click because I know of our time. So that's all going to do with hydrogen bonding. Tertiary structure, this is whole protein folding. So highlight that. These are far apart, but within the same sequence. So this is going to be because this one is hydrophobic and it folds inward. And this one is hydrophilic, and maybe it folds outward. So it exposes different parts of the sequence. And so then when they start to fold because of the polarity, you're allowing different interactions to occur. So we can again have ionic bonding, hydrogen bonding, or self-hydro bonding after that, that initial folding occurs. Okay, so this is making the one sequence into this big fat molecule. And there's one more level, because proteins are often not just one peptide, but they're several peptides. So the quaternary structure, so each of these different colors represents a different amino acid sequence. It goes through its initial folding, right? And then it is able to interact depending on what portion is showing on each of them. They'll have interactions that create certain shape as well. So this is when that's in the way. This is when the protein actually gets its function. So now we've reached its final structure. So like collagen is in our tendons, and our tendons need to be long, right, to connect between bones and muscles, and they need to be strong. And this reinforces that, and it's linear, so it allows it to do that. This, see how it has a tube in the middle? That is that same structure. Here you have groups where this is going to bond to substrates. So depending on the molecule, they might have different shapes for substrates. They might have different openings, things like that. So the quaternary structure, whole molecule, whole group, this is hydrophobic and hydrophilic hydrogen bonds, ionic bonds. Okay, so anything that affects primary also affects tertiary and quaternary. So the protein gets its final shape. That quaternary structure happens inside what's called a chaperone protein. So the chaperone protein here protects it from the solutions of the cytoplasm, because the solutions of the cytoplasm could cause their own ionic attractions, right, and pull the molecule apart. So the polypeptide has a molecule, ubiquin 9, that allows it to enter, and then each of the polypeptide chains would be in there, and so now they're held in close containment, you know, so now they're forced to have interactions with each other. And then when the protein is finished folding, it is released from that protein. So this acts like a giant enzyme, basically. So you can go through this. I just wanted to draw your attention to this. Like I said, you don't have to memorize which ones are polar and non-polar. If you can just identify the end piece, and from this you can't, but if it had a methyl group or a self-hydro group, if it had an amine or a carboxyl, if it had a carboxyl and donated hydrogen, if it had an amine and it tied up hydrogen. So it's recognize the pattern rather than memorizing each individual. And this is just a review. You got your primary structure, which is amino acid based on genes, based on DNA. Hydrogen bonds create alpha helixes or beta pleats. Hydrophobic, hydrophilic interactions allow for ionic and self-hydral attractions, which then are held in a chaperone protein that goes into, anyways, chaperone protein that then gives it its final structure. And so this is when it gets its functionality. Okay, so it is 8:24. So we've gone about, we're getting close to we've gone 40 minutes. I wanted to do this together, and then when you were done with this, you would click your turn in button. If you're anxious to get out of here, you are welcome. Hang on, let me jump to the end. Today I'm giving you this pattern matching. I told you about that already, and that's linked in the classroom. And it's just like a sorting. You grab and you sort. Okay, so if you want to work on this in here and then take off, you are welcome to do that, and I'll be here to answer questions that way. If you were like, man, I want to get out of here, then you are welcome to take off, do this on your own, and hit turn in when you're done. Okay, so if you're gonna hang out here, anytime you have a question that has a graphic on it, start with looking at the graphic and not looking at the questions and identifying what you know about the graphic to begin with, okay? So like if I saw this, I would right away know that I'm gonna be focusing on proteins. I know that this is my primary structure just because it's the sequence of amino acids. This is a secondary structure and it's an alpha helix. It looks like a secondary structure that is a beta pleat. So this looks like a tertiary structure because it only has one pattern. So that makes me think it's one amino acid sequence. And this has two different patterns. So I know that's going to be two different sequences. So this is my whole group fold. So first, identify what you know before you ever look at questions. I know secondary structures are based on hydrogen bonds. I know tertiary are hydrogen, ionic, hydrophobic, sulfhydryl. I know quaternary is also ionic, sulfhydryl, and hydrophobic. Globular is like a globe. So you can have filamentous, which is long, like a collagen fiber, and you can have globular protein. So like hemoglobin is literally telling you its classification of proteins is a globular protein. Okay, and then this one, I see a primary structure and then it's folding. So the black colored amino acids are the hydrophobic ones because they're found on the inside, right? And they're gonna orientate themselves away from the water. The lighter color is the hydrophilic portion. So when it's exposed to temperatures outside its normal range, it denatures. So remember, molecules move more quickly when it's hot and slowly when it's cold. So hydrogen bonds get disrupted. That was part of what makes water a liquid and ice solid, right? The density portion. So molecules move more quickly when they're heated, which disrupts hydrogen bonding, which is what ultimately causes the protein to fall apart. By denaturing it, you've changed its shape and it can no longer do its function. Okay. So on that, just go ahead and click done on your Google Classroom. So that moves out of the list of things for you to do. Sorry, my chat box, like I have like 4 windows open when I run the Zoom and I often miss my chat box.
Wait, Miss Snook.
Yeah.
Which one is it that we clicked done on? Like what's the...
Today's Zoom.
Mark is done.
Oh, one that says Friday Zoom? Or, oh wait, that's tomorrow. Is that for today?
I'm coming. Okay. So I tried to put, do you have the same order of things when, like, because I can click and drag them into different orders? No, so it's just my screen. Okay. Zoom time. Okay, I put the wrong day. It's Thursday. I just get lost. So much to do, that one. Because we had such a big list to go through today. And so we spent a bit of time just kind of wrapping our heads around where we are. So just go ahead and click, I'll edit that to say Thursday. And I know I said tomorrow at 3:30, but like this is our attendance check. So the quicker it's in, the quicker I can do attendance and